Hello everyone, welcome to the channel Microbial World. Today we will be discussing about endospore staining. So first we need to know what are endospores. Bacterial endospores are metabolically inactive, highly resistant structures produced by some bacteria as a defensive strategy against unfavorable environmental conditions. So a normal bacterial cell is known as the vegetative form of the bacterial cell. Okay. When they are provided with favorable environmental conditions like nutrients, temperature, water and oxygen, the bacteria will multiply freely. But some group of bacteria, when they are exposed to unfavorable environmental conditions like lack of nutrients, insufficient supply of oxygen and carbon dioxide, lack of water and moisture, okay, so during these conditions, the bacteria will form a protective covering to protect themselves from these harsh environmental conditions. And this special structure is known as bacterial endospores. So the endospore is a non-vegetative structure produced by a group of bacteria belonging to the Firmicute family. So why the endospores are considered a non-vegetative structure? It is because they are not actively involved in the metabolic activities of the bacterial cell. And we also said they are inactive, highly resistant structures, right? They are resistant to high temperature, radiation, chemicals like disinfectant and acid. Okay? And the bacteria can remain in the suspended state until conditions become favorable. And when the conditions are favorable, they can germinate and return to their vegetative state. Endospore staining. In 1922, Dorner published a method for staining endospores. Schaeffer and Fulton modified Dorner's method in 1933 to make the process faster. Endospore stain. It is a differential stain which selectively stains bacterial endospores. Okay? And what is the purpose of endospore staining? It is to differentiate bacterial spores from other vegetative cells and to differentiate spore formers from non-spore formers. Endospore stain reagents. So based on the type of reagents used, endospore staining techniques is classified into Schaeffer-Fulton stain that uses malachite green dye, which is the primary stain, and safranin, which is the secondary stain. Dorner method of endospore staining uses carbolfuchsin stain, acid alcohol and nigrosin. So in this video, we are going to discuss about Schaeffer-Fulton endospore staining method. Now let's see the principle of Schaeffer-Fulton endospore staining method. It is the simplest method used in basic laboratories because it makes the staining process simple and we can quickly identify the bacteria. And as we said, malachite green dye is the primary stain that is used. It is a water-soluble dye with an alkaline pH range of 11 to 12 and it combines steam heat to soften the endospore coating and allow dye penetration into the spore. So why it is important to provide steam heat? It is because this primary dye, it is a weakly binding dye to the spore wall, okay? And it will be washed away easily when rinsed with water without fixing. So steaming will help malachite green to permeate the spore wall. That is the reason why it is important to apply steam heat. Decolorizing agent water is used to wash the malachite dye from vegetative forms. So water will wash the malachite dye from the vegetative cells, but it will not wash the dye from the endospores. Okay? And then the decolorized vegetative forms are stained with the counter stain, which is also called the secondary stain, that is safranin reagent. So at the end of the staining process, the vegetative cells will take the color of the counter stain and will appear pink in color, and endospores will take the color of the primary stain and will appear dark green in color. Spores can be located in the middle of the cell, at the end of the cell, or between the end and middle of the cell. Spore shape is of diagnostic use and it may be spherical or elliptical. Procedure of spore staining. Take a clean grease-free slide and make smear using sterile technique. Air dry and heat fix the organism on a glass slide. Flood smear with malachite green and place on top of a beaker of water sitting on a warm hot plate, allowing the preparation to steam for 2 to 3 minutes. So one thing you need to be careful is that do not allow the stain to evaporate. And also you need to prevent the stain from boiling by adjusting the hot plate temperature. Remove the slide from the hot plate, cool and wash under running tap water. Counterstain with safranin for 30 seconds. Then again wash with tap water. Blot dry and examine the slide under microscope for the presence of endospores. So as you can see, first the primary stain malachite green is used and then steaming is done for 5 minutes, right? So all the vegetative cells and the endospores are green in color. When the decolorizing agent water is used, all the vegetative cells will be decolorized, but the endospores are not decolorized and they will retain the green color. Later, when the counterstain safranin is used, the vegetative cells will take the color of the counterstain and they appear pink in color, whereas the endospores are green in color. So the result of staining is the vegetative forms will take up the pink or red stain from safranin, while the endospores will stain green from the malachite green dye. Examples of spore-forming bacteria are Clostridium perfringens, Clostridium botulinum, Clostridium tetani, Bacillus anthracis, Bacillus cereus, Desulfotomaculum species, Sporolactobacillus species, Sporosarcina species, etc. So this is all about spore staining. Thank you.